So we will wrap up. I kind of said I was going to skip over these slides, but I can't make myself do that. So we will finish. Seriously, there we go. So kind of where we ended was talking about tertiary structure and what drives protein folding. What's important for protein folding and tertiary structure. So the hydrophobic effect. And who can kind of describe what we mean by the hydrophobic effects?
Just when proteins fold, like certain ones with certain side chains are hydrophobic or the inside.
Right. So as we practiced with our alpha helices on Monday, those alpha helices, so that if we have an amphipathic helix, the side of the helix that's hydrophobic would be more on the interior of the protein. That side that's hydrophilic would be on the outside of the protein, the exterior, exposed to solvent. So as a protein is synthesized on the ribosome, it folds up spontaneously. We'll talk about that in a minute. It's kind of crazy that the protein knows how it's supposed to fold, and really how it starts folding is it collapses to get those hydrophobic residues pointing to the inside of the protein. Okay, so it's not ever really going to be a stretched out linear chain. Even as it's being synthesized on the ribosome, it'll start to fold up with the hydrophobic side chains being buried on the inside of the protein. So when we're going to talk about quaternary structure in a minute, if you have two pieces of a protein, two separate polypeptide chains, they're going to form a dimer. Often if you can see the surface of one of those monomer units having a hydrophobic patch or an area where you have hydrophobic residues, that might hint that that's where the two monomers come together. Because in solution, you wouldn't want to have a hydrophobic patch. Leucines, phenylalanines, what else? Isoleucine on the surface of the protein. So maybe if those two dimers come together, that hydrophobic patch will get buried. So we've already talked about hydrogen bonding. And so hydrogen bonding is most important for secondary structure, right? So the kinds of hydrogen bonds that can form, whether they're local, so that would indicate more of an alpha helix. If they're bringing parts of the structure far apart, that might be a beta sheet. So those hydrogen bonds will fine-tune the structure, but really they're not forcing the protein to fold up. So hydrophobic effect drives protein folding overall, and then the hydrogen bonding kind of fine-tunes that structure. So it will help decide whether a certain region of protein should be an alpha helix or a beta sheet or a turn, for example. And the reason for that is if you have any amino acid or the nitrogen amine in the backbone, so if it's not forming a hydrogen bond to another part of the protein, what could it form a hydrogen bond to? Water. Okay? So it's not like you're winning or losing in hydrogen bonding, it's just which hydrogen bond gets made. Okay, so there's always plenty of things to hydrogen bond to in a protein. If it's not another part of the protein, it's the water. So that's not really, as we're counting up energetic benefit of folding the protein, hydrogen bonding isn't really a big benefit. Okay. And this idea of salt bridges. What do you think we mean by salt bridges? What amino acids could make a salt bridge? So did I hear lysine? Glutamic acid. Okay, so a positively charged side chain could, on the inside of a protein, make an electrostatic interaction. So lysine to glutamic acid, for example. A plus charge to a minus charge. So sometimes, people who study structure hate that term because it's really not a bridge. It's an electrostatic interaction, but we still tend to call them salt bridges. Okay. Okay, so we're going to end our protein structure kind of description by talking about quaternary structure, and that's the last level of protein structure, and that just means subunits of proteins coming together. So an individual subunit, like we've already talked about hemoglobin, we'll talk a lot more about it. So any of those monomer units, that's tertiary structure. As those monomers come together, that's quaternary structure. So it just means the fourth level of structure. So not every protein has quaternary structure. Certainly plenty of them are just a single polypeptide chain that folds up and is functional. Again, we already said that the surface of those interactions may be hydrophobic interactions. There certainly can be hydrogen bonds. There certainly can be disulfides, covalently cross-linking those, but that's pretty untypical. Usually they assemble by non-covalent assembly. So there's nothing covalently holding them together. So how do you think we would get subunits of a protein with quaternary structure apart?
Yes?
Chris? Okay, so some kind of detergent that might denature the protein, and maybe even a lower concentration of detergent would take the subunits apart while keeping the tertiary structure of each subunit intact. How else maybe? Maybe just a little bit of heat. Okay, so if it's primarily either hydrophobic or hydrogen bonds, those are weak forces, and so maybe if we just heat it up a little, they'll come apart. Okay, we'll also, so those of you who are taking lab, you'll also do denaturing gels. So that's definitely a detergent. That's definitely other denaturants that will totally unwind the protein, and then if it's unwound, the structural parts can't interact with each other, so then the subunits definitely come apart. Okay, here's just some examples of quaternary structure, and the different colors represent the different subunits. So here we have a dimer with a plane of symmetry. Some of these actually are not so much mirror planes of symmetry, but rotational symmetry. So if you take that HIV aspartic protease and rotate it 180 degrees, the subunits will, the pink subunit will now be where the purple subunit was. Okay, so many quaternary structured proteins have rotational symmetry. And here's a protein with trimeric symmetry, and again, the rotational axis is straight up and down, or coming out at you, I guess. And here we have a tetramer with that kind of rotational symmetry axis. You don't need to know about symmetry, just that those proteins can have different kinds of organization. And go from simple dimers, trimers, tetramers to very complex types of symmetry. Often viruses, their protein coats will have more complex kinds of symmetry. So what does icosahedral, how many subunits is that? 20? Yeah. 20 subunits. Okay, and so viruses typically have the proteins on the outside protecting the genetic material on the inside. So it's kind of like the candy coating on an M&M. You don't want the chocolate exposed to the warm atmosphere, so you can cover it with a candy that will protect it. Same thing for viruses. So they're either DNA or RNA on the inside, and the protein is the protective shell that also triggers interaction with cell types, etc. And so if we could isolate just one of these protein subunits, what do you think we could do with it? If you just had one protein copy from the viral coat, what do you think you might do with it medically? Say that again? You could duplicate it. You could use it as a vaccine. Okay, so polio virus. So the first vaccines against polio, who developed those? Who came up with that idea? So who? So Salk, so certainly made them better. So who had the first idea of inoculating people? Yes. Pasteur, very good. Okay, so Louis Pasteur kind of thought, ooh, how could we protect people? He looked at different diseases in chickens and other animals, and then was able to inoculate people by essentially diluting out the live virus. And so Sabin and Salk, you're right, Andy, were the kind of competitors of, do you kill the virus or do you just dilute it? And so if you have a very dilute virus that you give as an antigen to raise antibodies, that's a little dangerous, right? So these days, almost all vaccines are what are called recombinant vaccines, where you just express one of the coat proteins and raise antibodies against the coat protein. So if you have an antibody that will recognize the protein on the surface, now your immune system so if you raise antibodies in your immune system against the coat protein, now if it sees the whole virus, it should recognize and eliminate that. Okay, so you kind of get around the putting real viruses into people. Yes? That would make the most sense. And so that's the difference between monoclonal and polyclonal. So if you have a polyclonal antibody that's multiple antigens, probably different coat proteins, maybe even different strains of the virus. So certainly the flu vaccine, so every year they decide, okay, which ones are you going to combine in the vaccine to try to give the greatest protection, because you have slight differences on the surface. Okay, so if we have these structures, and we're going to talk in a minute about how we know what the structures look like. So I already said the protein knows how, from the sequence, to fold up and make a structure. So is it going to be a coat protein structure? Is it going to be hemoglobin? Is it going to be what? So the protein knows the structure it's supposed to be. Do we know from sequence how to predict protein structure? So the answer really is no. So we can look at a DNA sequence and know what protein it's going to be. We can't look at a protein sequence and know what structure it's going to be. Okay, so lots of very smart people, lots of computational biophysicists are trying to figure out what the secret hidden code is. So researchers back in the 60s figured out the genetic code, DNA to RNA to proteins. The structure code is still pretty mysterious. So looking at the sequence of amino acids in a protein, we can't de novo, or from nothing, predict structure. But if you have a whole series of different proteins and know their structures, now the closer a sequence is to one of the sequences that's already solved, the better the guess is about structure. Okay, so if it's hemoglobin from whales versus hemoglobin from people, probably they're going to be pretty similar. But hemoglobin versus some other random protein, we can't tell just from the sequence. So we're not very good at that. So this is homology modeling. So if you have a known sequence and you can compare the structure, then you can make predictions that still have to be tested. Okay, so again, this is the idea of protein folding. So we know it's spontaneous, but how does it happen? So if you had a protein of 100 amino acids, how long do you think it would take that protein to fold? Do you think the protein has to try out all different combinations, all phi-psi angles? No. So it does happen spontaneously. It happens very quickly. So does anyone know how fast amino acids are added to a peptide chain? So how fast does protein synthesis happen? So most people would use numbers like 40 amino acids per second. Okay, so that's very fast. If you think about all the things that have to happen, decoding the message, making the peptide bond, kicking off the empty tRNA, moving everything along. So 40 amino acids per second is very fast. And if the protein folds up as it's being made, folding is very fast too. And so it's spontaneous. So somehow there's a hidden code in the sequence that we don't know what it is. And it's not random, and it's very fast. Okay, so again, this is the idea of the protein structure code. So if a protein had to try every phi-psi angle, the universe isn't old enough for that to happen. So again, that points back to the structure code. What about reversibility? So protein folding is, in most cases, reversible. So if that protein folds up spontaneously, even as it's being made, so this is an example of ribonuclease. So what do you think ribonuclease does as an enzyme? From the name, what kind of hints? Cleaves RNA. Yep, it's not that fancy. So ribonuclease is a small protein. So what's that, 120, 130 amino acids. So it's a compact, small protein. It has 4 disulfide bonds. So the yellow things are disulfides. Be careful not to touch what I'm not supposed to touch. So those are disulfides. So how would we break up that protein, okay, to get it to its fully stretched out version? So urea is a compound that we use. So urea is this. So it's a denaturant. So it will separate the protein, disrupt the hydrophobic interactions, and allow it to come unwound, basically. And mercaptoethanol. Does anyone remember that we talked about that before? So it breaks the disulfide bonds. So it's a reducing agent. So mercaptoethanol or, so 2-mercaptoethanol or beta-mercaptoethanol or DTT dithiothreatol is another separate compound, but those are all reducing agents that will now break those disulfide bonds. So now we have 3 thiol groups on the cysteines, so the whole thing has come apart. How do you think we get it to fold back? So how would we get rid of those chemicals that just allowed us to unfold everything? So we would have to dialyze away those chemicals. Okay, so in lab, if you're in lab, you will use dialysis bags or dialysis tubing. You can also kind of filter, but that is a little bit more complicated. So if you can get rid of the urea and the mercaptoethanol, it turns out that the protein folds back spontaneously and makes the same disulfide bonds it made on the ribosome initially. So it doesn't mix and match the disulfide bonds. So again, the sequence knows what the structure is supposed to be, which disulfide bonds are supposed to form. So not only is it structurally intact, it's functional. Okay, and so that was predicted from the beginning of what we knew about protein structure, but then confirmed back in the 1970s when this guy Christian Anfinsen won the Nobel Prize for experimentally confirming what people had predicted about reversibility. Yes?
Isn't there like a biological thing that unfolds if it's not folded correctly?
Yes, very good. So what are those called? So those are chaperone proteins, or what's the big complex called? I think I have a picture of it. So I always think of chaperone proteins or heat shock proteins. So the nucleosome is what chews up proteins that don't get folded. So definitely protein folding is important, and it's a bad thing if it doesn't happen. So if we think about protein folding, again, it's important, it's spontaneous. Which parts of the protein are more likely to fold first? So if we have a choice of alpha helices or beta sheets, which ones should fold first as the protein is being made? Alpha helices. Why? Because they're closer together. So an alpha helix can be formed with a small, a stretch of polypeptide that's close together in sequence. Some beta sheets can do that, but many beta sheets are far apart in sequence and then fold up later on. So if we see, an alpha helix folding first, so first the protein kind of collapses to get the hydrophobic residues away from the water, and then the protein kind of says, oh, this part would make an alpha helix, that's handy, I can make more hydrogen bonds, and then you have the final folding up of beta sheets. So alpha helices fold first because they're more local, beta sheets are more distant. Okay, and so we know that when proteins don't fold right, that's problematic. So the two main kinds of things that can happen that result in diseases, we can get Alzheimer's disease, which is plaque formation in the brain. And so it's really just misfolded proteins that aggregate, or proteins that should get processed and destroyed, but collect and make basically precipitated protein in your brain, unfortunately. And then the other kind of misfolding, which is really wacky, are prion proteins. Okay, so this is mad cow disease, so that's our mad cow down there. So mad cow disease in humans, that's Creutzfeldt-Jakob syndrome. There's also a weird disease that only a handful of families have. That's also a prion disease, so misfolding, and it's called fatal familial insomnia. So what do you think the symptoms of that are? You can't sleep. So all of a sudden you hit 40, so they don't know exactly, what triggers it. It's definitely genetic. So all of a sudden you hit your 40s and you stop sleeping, and then you die because you never sleep again, and you go crazy, and just think like everything shuts down because sleep is necessary to, rest, kind of regroup brain function, and yeah. So apparently people need. There's like an Italian family, but by then you've had kids, you pass it on, So I think at this point they're catching on that maybe they want to remove themselves from the gene pool a little bit. But you kind of get to your 40s and you have one night where you had too much caffeine, and then they're like, oh, is this the end? So it's not 100% fatal in the families, but if you get it, you can't do anything. Yeah, I can't sleep, and then I'm going to die. But that is a protein that's supposed to be folded in one type of secondary structure, and some mutation just converts. So, for example, converts one structure to another structure, and then they kind of polymerize. And the bad protein kind of convinces the good protein to adopt the wrong structure. Okay, so you got one rotten apple of a protein, and all the proteins around it suddenly become rotten apples, and then the ones around that, and so it just goes downhill very quickly. The crazy thing is, it's not a DNA inherited disease. So Stanley Prusiner won the Nobel Prize not that long ago because he showed that there's no DNA genetic material that's passed on. Okay, and so that's why it was such a major big deal that if you ate hamburger meat in Great Britain in the 1980s, you can actually get Creutzfeldt-Jakob syndrome, or BSE, bovine serum encephalopathy, I think, whatever, by eating even hamburger meat that's been cooked. So by consuming the proteins, how it crosses the blood-brain barrier has not quite been figured out, but there's no, everything has been done to this meat to kill the DNA, break it up, denature it, and yet you can still inherit the disease. So that's like, this guy wasn't believed for many, many years because it's so antithetical to genetic diseases. Okay, so here's our chaperones. So this is kind of this assembly that gives proteins a second chance to fold. Okay, so if they're misfolded, let's put some energy in, let's unwind them all the way, and give them a protected space in the cell to try to fold up again. And then if they don't fold after a second chance and some ATP energy, then we're just going to chew them up and destroy them so they don't cause problems later on. Did I take that picture out, or is it missing somewhere? Oh, sorry about that. I guess I decided it wasn't important. I'm going to put it in anyway. Yes?
So all Irish drugs, are they all, like, just a chaperone?
No, unfortunately, I think most drugs against, so you can't really fix those proteins. So once they're in the brain, so most drugs that I'm aware of help cognitive function. So basically it prolongs your functionality as long as possible, but it's not fixing what's going on in the brain. So it's helping alertness or other things. And I guess I'd have to kind of figure out what that would be, but it's not, you can't fix the proteins in the brain, unfortunately. So I think it's more symptom relief than treating the disease. Okay, so we are going to talk a little bit at a very superficial level about determining protein structure. And so have any of you worked in a protein structure determination lab? Okay, last semester I had a couple. So there are two main methods for actually determining structure, are this one and that one, so NMR and X-ray crystallography. CD we're not going to talk about. That actually doesn't determine protein structure, it just allows us to compare structures. So it's useful in the lab, but we're going to skip it. It's fairly abstract, and if you want to know more about it at some other point, I can tell you if you want. Okay, so how many of you have done NMR in organic chemistry? Probably all of you. Good. So it's very useful if you have very small molecules. The bigger the molecule, the harder NMR is. And so if you think about a protein, we're not even talking about a dipeptide. We're talking about like 100 amino acids, even 50 amino acids. That's a problem. So if you get a signal for every protein in the if you get a signal for every proton in the molecule, how many protons are there in a protein? A lot. Yeah. Okay. So, and of course they're all going to sit right on top of each other. So every amide proton of the backbone, if you have 100 amino acid residues, you have 100 amide protons. So you have 100 signals in one of the peaks. Okay. So how are you going to deconvolute that? If you have 4 phenylalanine residues, you have 4 times 5 phenyl protons, and then they're all sitting on top of each other for the most part, or they're kind of a broad area. So how many protons are in that peak? Crazy lots. So how are you going to deconvolute that? So that's the challenge of using NMR for biological macromolecules. It's just not practical. So one-dimensional proton NMR like you would do for organic chemistry is just not even useful. So what do you have to do? So again, you're not going to know the details of this. I just want you to appreciate what's the problem and what might we have to do. So what we might have to do is do more sophisticated NMR techniques. So have you done kind of multi-dimensional NMR in organic? I think you do a little bit, like TOXI or COSY, or not, maybe not. So multi-dimensional NMR means we can't just look at the protons, we have to look at something else. So that something else could be C13. So of course there's lots of carbons in a protein. That would be great. So nitrogens. So where are the nitrogens in a protein? Well, all the amide nitrogens. But then you have nitrogens on lysines and arginines and glutamines and asparagines, but not on every amino acid. So the challenge for proteins is, you have all these signals, you have to identify which amino acid they came from and which functional part of the amino acid. So the identifying signals is a big challenge. So heteronuclear NMR is where you have not just the proton signal, but other spin-active nuclei that you're tracking, essentially. Homonuclear is where it's just protons that you're paying attention to, but you're doing things to the NMR to transfer energy between the protons. Okay, so that's homonuclear, same proton spin nucleus, but you're varying the radial pulses. So maybe you're going to put some energy in at a certain wavelength, and then you're going to watch it decay or watch it pass from one nucleus to another. Okay, so those are the tricks. They're sophisticated tricks that lots of smart people have figured out, what they do and what they mean and how to deconvolute, but that's the challenge. So you have to figure out which signal belongs to which amino acid and how are we going to learn which amino acids are connected and then what the structure is. So for the most part, if you have a protein you're studying by NMR, you know the sequence. So you would know what the connection is in the primary sequence. What you're trying to figure out is how is that primary sequence folded up into secondary, tertiary structure. So that's the challenge. So the only method I want to tell you about and have you try to understand, and I'll do my best, is what's called NOESY. So NMR people come up with wacky name, abbreviations for things. So there's TOPSY, there's NOESY, there's all kinds of weird names, and they all have to do with spectroscopy, right? So NOESY is nuclear Overhauser effect spectroscopy, and what it probes is atoms, protons, that are within 5 angstroms in space. Okay, so there are 2 ways to transmit energy in molecules by NMR. One is through bond. Okay, so if there are 3 bonds or fewer, that energy will pass, and the other is through space. So those are both energy transfer kind of methodologies. NOESY, nuclear Overhauser effect spectroscopy, monitors through space energy transfer. Okay, so the only way that you would get a signal is if you have protons that are within 5 angstroms of each other in space. So if you knew, so again, you still have to identify which proton is coming from which amino acid, but say you had a proton on amino acid 2, and it was passing its energy to a proton on amino acid 33. So what would that say about those amino acids? They're close in space. So however, whatever the backbone does, they have to end up close in space. Okay, and so as you begin to collect information about lots of different protons that are connected in space, less than 5 angstroms, you can computationally model what does the backbone have to do to meet all of those space constraints. Okay, so NOESY, just as one example, is going to tell us which protons are within 5 angstroms in space, and we're going to use those distance measurements or distance constraints to come up with models of how the protein must be folded. So NMR doesn't give us one structure, it gives us lots of energetically favorable models that fit the data. Does that make sense? When we talk about X-ray crystallography, that actually shows us the picture. Okay, so the through space spin transfer is important. So if we just look at the peptide kind of schematically, so this is just the naming. Okay, so this is, and these are all protons. The only signal we get are protons because it's homonuclear NMR, just proton. So if we have a signal between the proton on the amide of residue 1 and then the amide of residue 2, that's what we call that signal. Okay, so it's just, they're classifying what they are. Okay, and so there's only so many that you can get that are within 5 angstroms in space or 3 bonds. Okay, and so what's the comparison? What's the analogy? Yes?
Could you go back to last slide?
Mm-hmm.
So why are some of those
Well, so this is just the backbone, essentially, the C alpha, what's on the C alpha.
Okay, but that hydrogen still, like, affects like the other hydrogen proton.
So this is just kind of showing you what they're calling them. Okay, so I guess the D is just the transfer. It's not really a distance, but the beta, so that's the proton on the C beta. So this is the C alpha. The next carbon in the side chain would be the C beta. The next one would be the C gamma. So we're just kind of naming that proton beta. Okay, and we're naming this one N. Okay, and that one is alpha. Yeah, so it's just kind of how do we keep track of what they are. Okay, so the analogy is, back when we used to use maps and atlases, does anyone Has anyone ever looked at an atlas? Okay, so they're actually kind of cool. So my son in 5th grade, like for geography, one of the hints on practicing geography is when you travel, don't just use the GPS, break out a map and kind of show, distances and how things are arranged relative to one another. So at the back of an atlas, you used to be able to find distances between cities. Okay, so Albany, New York to Albuquerque, New Mexico, almost 2,100 miles. So if these are all distance constraints, so how far those cities are away from each other, if you had enough of those, you could reproduce the map of the U.S., right? So how would you have to arrange all those cities so that all of those distance constraints could be met? Same thing for protein structure, except now instead of a 2-dimensional map, we have a 3-dimensional map of where all the protons are. So if we know all the protons, and there could be thousands of protons in a protein, if we know all the protons that are within 5 angstroms of each other, how would we have to fold up the backbone in 3 dimensions? How would the side chains have to be rotated to meet or satisfy all those distance constraints? Does that make sense? Okay. And I know this is very abstract. So if we have an alpha helix, what would that look like on a NOESY data set? Okay, and so this is kind of how we represent them schematically. It's not real data, it's just kind of a summary of the data. So if in an alpha helix, we've talked about hydrogen bonding between the carbonyl oxygen of residue I, or N, or whatever we want to call it, and I plus 4, so, and the amide proton of residue I plus 4. So we talked about those as hydrogen bonds, so between a proton and an electronegative atom. The comparison is, in NMR, we're only looking at the protons. Okay, so it's not between the carbonyl oxygen and the proton, it's between a proton and a proton in the peptide. So for an alpha helix, the strongest NOE signal is between the proton on the alpha carbon, so alpha is the proton on the alpha carbon of residue I, and the beta proton on residue I plus 3. So don't get freaked out by the I and I plus 4, because that's a hydrogen bond, which is different than the protons. Okay, and I see I just lost you. So if we call this residue 1, 2, 3, 4, okay, so that hydrogen bond is between the carbonyl oxygen and the amide proton of I to I plus 4. For the NOE, so the proton signals, okay, so if they're alpha, so can you see, let me change colors here. Let's see if that helps. So can you see this proton, so that's on the C alpha, and we would have that C beta proton, okay? So because for a hydrogen bond, the carboxyl oxygen is kind of on the C prime end, sorry, the C end of the amino acid, and the amide proton is on the N end. When we look at the hydrogens, the protons for NOEs, the numbering is just a little different. Okay? But, so the darkness of the lines over here indicates how strong that NOE signal is. And so the stronger the signal, the closer they are in space. So if you see nothing, that means they're more than 5 angstroms away. If you see weak lines, that means they're within 5 angstroms. And if you see it darker, that's the closest they get. So the closer they are, the stronger the signal. And so the strongest signal is between the C alpha proton of one residue and the C beta proton of the residue I plus 3. Okay, so bottom line, that stair-step pattern indicates an alpha helix, because you don't just have one of those proton-proton distances. The next one down also has that, and the next one down, just like the hydrogen bonds. So you're not measuring the hydrogen bonds, but it's kind of duplicated in that stair-step pattern. Does that make sense? Questions about that?
How would you identify a beta?
Okay, so a beta sheet, thank you, doesn't have that. Okay, so you can ignore, so this is just a slightly different kind of helix. What we care about is alpha helix, and this is a beta sheet. So for a beta sheet, what does the backbone look like? Fully stretched out, okay? So are there any signals between residues I and I plus 3, or I and I plus 2, or I and I plus 4. No, because they're far apart in space. Okay, so the only signals you get for a beta sheet are the protons that are next to each other in the sequence. Okay, so these are near in sequence. So of course they're near in space. And you're missing all the long distance, both hydrogen bonds and protons through space
It
connections.
satisfies the no-scope, so it doesn't do like I plus 5.
So after you get to I plus 5, they're too far apart. Yep, yep. Okay? So the strongest NOEs for an alpha helix are between those protons, and the weaker ones are just the ones around. Okay, so that's just kind of the fingerprint of an alpha helix versus a beta sheet by NMR. So what's great about NMR? If it's so much work to identify what the amino acids are, do all these sophisticated NMR tools, you need a big magnet, bigger than what we have in fact, so why would anyone ever do this? So there are advantages in that you don't have to crystallize a protein. So when we talk about protein crystallography, that's kind of a big hurdle and not always possible. So we're going to see in protein, so most of these advantages are in contrast to crystallography. So if something's hard in crystallography, sometimes you can get around it by NMR. So it's an aqueous sample. You do need high concentrations. Sometimes you can use NMR for membrane proteins. So what do you think is the challenge for membrane proteins to try to crystallize them? So membrane proteins are typically in a nonpolar lipid environment. When we see crystallography, we often use aqueous solvents. So it's just hard to get them solubilized. So sometimes by NMR you can use tricks and put them in micelles and lipids to actually use NMR in a lipid-like environment. So they're great for small molecules and for dynamics. So that means if you have identified which protons are from which amino acid, now you bind a ligand or you change the environment, you can see what moves. So what's different under this state versus this state? It's an aqueous sample. You can just look for the spots that move, and that's much easier. A couple of Nobel Prizes. Those are for applying kind of organic chemistry NMR tools to bigger molecules. So what are these tricks? What are these nosy kinds of tools? Okay, and we will start a little bit of crystallography. So back when we had space shuttles going up, sometimes they would take proteins to crystallize. So we'll skip that, except just to say that in order to crystallize a protein, sometimes low gravity helps. So once when I was in grad school, I heard a talk by an astronaut scientist. It was like, it was the best experience of my life, but I'm basically going to be paying for it physically for the rest of my life. So once you're in a low gravity environment, so he said most astronauts have back problems the rest of their lives because you're kind of stretched out in low gravity and then you kind of compress and the spine just doesn't like it too much. Okay, so I'm going to say all that kind of graphically here. So the challenges for X-ray crystallography, the steps, which again you're not going to do probably, maybe you will. You have to crystallize a protein. So this is basically in a nutshell. Going from crystallizing a protein to getting electron density to what does the electron density look like, and then how do we fit the protein sequence into that. So this is basically the equation you solve. Easy, right? Yeah, so that's the hard part. So X-ray crystallography, back in the days of Jane and David Richardson and Rosalind Franklin and her friends, it was a big, brain-heavy endeavor. Okay, lots of paper and pencil, slide rulers, all kinds of heavy mathematical manipulations that you would have to do kind of by hand, slide rules, etc. So how do you think we do a lot of X-ray crystallography now? Computationally. Okay, so there's lots of computer programs that will take the data and kind of by trial and error, iterative process. Does the protein backbone fit in this form? No, not quite. Let's tweak this. So rather than solving the equation, it's more like fitting the backbone into the data and kind of predicting forward. So I will end there and see if anybody has any questions, and then we'll wrap up with protein crystallography, the cool part of making crystals on Friday. Any questions on amino acids, protein structure?
Would this be like the last step that we could cover, or would it just be
So we will, so whatever we get to on Friday, will be on the exam, but not Monday. Okay? So the material on Monday will not be on Wednesday's exam. So hopefully by the end of Friday, so we're just going to kind of zip through protein crystallography. I'm too nice to do it in 2 minutes, but there's, it's mostly just appreciation for the effort that it is. Then we will talk about protein function, hemoglobin, oxygen binding, ligand binding, so a little bit of calculation there. I think most of you have been exposed to hemoglobin. We're just going to dig into the more chemical atomic aspects of it. Okay? Okay. Yes?
Residues are in 5 angstroms, like approximately.
5 angstroms of an alpha helix.
Yeah.
So if it's about 1.5 angstroms along the alpha helix, so what, 2.5? Right? So if it's 3.6 angstroms per turn, and that's 5, so that's actually 5.4 angstroms, right?
Yeah.
Yeah. So it's about 1.5 angstroms per residue.
Okay.
Yeah, so 5.4 is 3.6, so like 3-ish. Yeah, because 1.5, 3, 4.5.
I just had a question. So, I understand I got off for here, and
So it looks like
so
you...
because I averaged these two.
Yeah, that's not very good.
Okay, so the main thing is you just look at the residues that are actually there, and then whatever get in between it or keep her.
Right, so this was right.
Right, and then
because
I
of course leucine doesn't have...
Yeah, it doesn't have. It only has this amino.
Right,
And
so-
it doesn't, yeah.
Right, so essentially, when you make the dipeptide, the only contributors are the carboxy terminus,
Okay.
which came from the histidine
Right.
side chain of the residue, the ammonium terminus, which actually came from the